So I call them genetic testing, but it's kind of, whatever you want to call it. I call them genetic approaches because they are dealing with the nucleic acids, that's all. So the first approach here will be, I told you, you heard this term, PCR. Okay? Does that make sense? Polymerase chain reaction. I debated with myself whether or not we should go step by step in a really painful way and decided not to. But I wanted with you to look at sort of the outcome or applicability of the test. So what essentially PCR does, it uses certain reagents like DNA polymerase and primer. So basically it uses nucleotides to recognize a short fragment of the DNA of a microorganism. Does that make sense? So let's go back to our beloved coronavirus. We can design reagents. They are design reagents. We can design them so they will be so specific that they will bind only to the coronavirus genome. Only, nothing else in the entire world. Does that make sense? So they will be able to recognize certain sequences on the, I use the term coronavirus genome and I'm cringing because technically speaking it's incorrect. We have to convert RNA genome of coronavirus into DNA. It can be done, it's technicality. So I'm going to be using genome, but we'll remember it's a little bit more complicated, not important for our purposes. So those reagents recognize particular sequences in the genome, okay? And due to that recognition, your PCR toolkit can amplify this one copy of the genome of that particular fragment, not the whole genome, but that particular fragment of coronavirus genome over and over again. This is how it works. First round of reactions, you get 2 copies. Does that make sense? Second round of reactions, you get 4 copies. Now, anyone's guess after the third round how many copies we're going to have? Doubles. So 8, and then 16, and then 32. To make a long story short, after 30 cycles, you're going to multiply the original number of the genomes more than 1 billion times. Does that make sense? Any questions so far? So let's start with the sensitivity. So how sensitive this method? Technically speaking, you can determine a single copy, one genome in the sample, which is mind-blowing, really. Okay, that makes sense? Second thing, and now I'm going to get to a real practical application to the coronavirus story. So everybody knows some places, like some countries, required negative PCR test, right? So what does that mean? The test is negative. When does it negative? So according to CDC guidelines, well, before CDC guidelines, let's figure out another thing. Can you make this test quantitative? The answer is pretty much yes. What you can do, you can set a particular threshold, okay? I can't put it on the board. So you can set a particular threshold, and then you can count how many cycles, you see? How many cycles it takes to reach that threshold. Are you following me? So if you have 100 copies in the beginning, it's going to take less steps to reach a threshold compared to if you have just one. Because you start with more material. Does that make sense? So CDC, based on epidemiological data, sets the threshold at 36, which means if in your sample it took more than 36 steps, 36 cycles, to get that particular threshold, you're negative. If it took less than 36, you're positive. Which begs another question. What if it took 35? You probably are negative. Moreover, what if it took 32? Are you positive? According to CDC, you are. Are you infectious? Based on all the epidemiology that we have, you aren't. Does that make sense? So this is kind of, something that I just, everybody should keep in mind when you're being told you're negative or you're positive, what it really means. Does that make sense? Again, one of the downsides to PCR is that whatever you have primers to, like if you have a patient who is dying of pneumonia and this patient is coronavirus negative, all it tells you, it's not coronavirus. It doesn't tell you what it is. Make sense? To be completely honest, there are respiratory PCR panels in the hospitals which test for like 18 different respiratory pathogens at the same time. It's very complicated. I mean, the setup is not really, but like the reagents and stuff. But you can do that. It's called multiplex PCR. But yeah, it's still, you are limited. Like if it's not among those 18 respiratory pathogens, you're still without answer. Make sense? Plus side, relatively fast, relatively cheap, very easy to automate. You can process thousands of samples a day. Now, third method, which is one of my favorites, I'm going to put, the only thing that I'm going to put about this method on the board is the name. It's called sequencing. Okay? This is why I put only sequencing. So original methods like Sanger sequencing and Maxim Gilbert sequencing were very tedious, resource intensive, and so on and so forth. The modern sequencing allows you to determine a full sequence of DNA or RNA in the sample. Does that make sense? I tried to understand the mechanics of it. I almost did, but in the process I realized that people who developed it were smoking really heavy weed, like really heavy, because you can't come up with this stuff like being normal, being sober. It's very technically complex. So let's jump to the results right away. Here's what happens. You take a sample and put it in the magical machine. And magical machine gives you the sequence of all DNA, everything that is in your sample. So if we go back to that patient, and we would put the sample of the lung properly prepared into the machine, we would get patient samples, probably some bacteria that accidentally were in the lungs, coronavirus, all that kind of stuff. So this is good and bad. Good because you get all the information possible. You have everything. Bad because you have all the information possible. And you have to swim in that ocean of sequences trying to figure out what's going on. Like, what is the culprit? Does that make sense? Questions? Okay. What we're going to do